Hello, everybody, and welcome to our webinar today. We are still having some people trickle in, but we're going to get started just to try to keep on time. I know many of you are joining us during your lunch break or eating while listening, which is totally okay. We will do most of the talking for the beginning of the webinar and then go through a question and answer at the end. We have a couple people who will be sharing a little bit of information today, so I want to just start off by introducing ourselves quickly. My name is Brittany Rogers. I am the Aquatic Restoration and Resiliency Coordinator with SLELO PRISM, or the St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management. We use the acronym obviously because it's a little bit shorter, but thank you so much for joining. And Mary and Pippa, if one of you would like to unmute and share an introduction.
Sure. Hi, my name is Pippa. I also go by Philippa, that's my full name. And I work at the Nature Conservancy out of Rochester, New York. And I am a sustainable fisheries ecologist with TNC.
Hey everyone, my name is Mary Ripka. I'm the New York State Volunteer Programs Manager, and I'm based out of the Pulaski office in Northern New York.
Thank you so much. And thank you again to everyone on the line. Some of you have found the chat already, so I'm happy to hear that. But if you would like to introduce yourselves in the chat or message any of us, you can at any point in time during this webinar. So with that, we will introduce you to what we're talking about today. Pippa.
Great. So thank you all for attending. And like Brittany said, we realize this is your lunch break. So this is wonderful that you have decided to spend it with us. So today we're just going to give you guys a brief overview of who we are as an organization. So this includes the Nature Conservancy, as well as the SLELO PRISM that Brittany mentioned. We're also going to provide a bit more detail on the specific project that we are embarking on. We're going to provide a little bit of information from our funder, the Archonic Foundation. And then as Brittany mentioned, we'll have time at the end for any questions that you guys may have.
Awesome, thank you. And I hope everyone can hear my audio okay. My computer did freeze earlier, so I am connected with two devices to try to prevent any technical difficulties. But with any webinar, I'm sure we will experience something at some point in time. We will be keeping everybody on the line muted, at least while we're doing the initial presentation. And at the end, as Pippa mentioned, that Q&A, if anyone wants to unmute, you can, or if ask questions in the chat box, we can do that as well. Just so some brief housekeeping when we get started, I just want to introduce you to Zoom. If you haven't been on a Zoom webinar before, you can probably see a similar screen to what I'm showing you here today. And you can open and close your participant list and the chat box by clicking on the bottom of your screen there. If you're having any issues with audio, or for some reason something seems a little bit off, you can click that little caret that points upwards next to the unmute button, and then check your microphone and your speakers. And you can also test your audio as well here. If you find that you'd rather call in and leave your computer audio, you can do that. Just make sure you don't have two audios going at the same time. You might get a little bit of an echo. I see many of you have joined us with video, which is fantastic. I would love to see everyone in person. Obviously, that's not necessarily something we can still do right now, but I'm glad that we can all join from all over the state. And there's even a couple people from out of state that have joined us. If you want to turn your camera on or off, you can click that caret button or start and stop your video there as well. And then finally, in the chat box, you can see there is an opportunity to choose your messaging, but then also when we get to the Q&A part, you can raise your hand by clicking that little raise hand button or any of the other information. I know I'm a fast talker and I get excited about some of the work that we're sharing. So if you find that we're moving a little bit too fast, or if you have questions, you can send those in the chat or we can clarify things towards the end. I'd like to at least initiate this webinar by first recognizing how important it is to understand the longstanding history that has actually brought us to reside on the lands that we live on and work on and enjoy. We have spent a lot of time trying to better understand our history and our place within that history. And when we talk about lands and waters like we're going to today, we have to acknowledge that it is a part of who we are. It's a mixture of our blood, our past, our current, and our future. And we're hoping that projects like this will help us continue moving into the future to be better stewards of the land. We do need to protect and honor the history and many of the people and places where this project will actually fall. And so I'd like to just take a moment to acknowledge some of the historical stewardship of the land and also the extensive history that our lands and waters have. And then also emphasize the need that we have to actually work together to also protect these important resources. So by looking at the map on your screen, you can see a plethora of areas that have over time transitioned between stewardship and who was actually residing on the land. And I'd like to at least bring to light the original stewards of the land. So I'd like to just briefly mention the Onondaga, Oneida, Haudenosaunee, and Mohawk nations. I know that a land acknowledgement simple like this is only one small part of supporting our Indigenous communities, and I hope that at least bringing this to your attention will inspire you to want to learn more about your lands and waters as well. If you want to view this map and also have access to some of the different nations within your region, you can visit that nativeland.ca. The link is at the bottom left-hand side, and I can also send that in the chat box for you later in the webinar. So thank you. And with that, I will mute myself and let Pippa take over.
Great. So as you mentioned, both Brittany and myself work for the Nature Conservancy. So I just wanted to provide a little bit of background as to who we are as an organization. So the Nature Conservancy, you'll also hear us call ourselves TNC, was actually formed in 1951 from a group of ecologists. And the very first chapter was in New York, actually, as well as their first land acquisition in the Mianus River Gorge. So we have a rich history in New York. And since that time, we have spread. So we are located or headquartered in Arlington, Virginia, and we have staff and offices worldwide. So we have grown and we are involved in various projects. Next slide, Brittany. So our mission is to conserve the lands and waters on which all life depends, and our vision is a world where the diversity of life thrives and people act to conserve nature for its own sake and its ability to fulfill our needs and enrich our lives. Next slide. So you may be thinking, okay, big organization, what type of things do they do? How do they kind of contain it all? So we have 4 priority areas. The first is tackling climate change. So this is thinking both about adaptation and mitigation. It's also protecting lands and waters. So you might think of us as a land organization, but we are actively involved in freshwater as well as the oceans. We also have a subset of work that's focused on providing food and water sustainably. And we have a whole program that's focused around building healthy cities. Next slide, please. So in New York itself, we have kind of 4 main areas or the way of dividing up the state. So both Brittany and myself sit in central and western New York. We also have folks up in the Adirondacks. As I mentioned, our first acquisition was in the Mianus River Gorge. So we have numerous preserves and staff in the Hudson Valley. And we also have an office in New York City and on Long Island and a really cool preserve on Mashomick. Next, please. And now I'll turn you over to Mary.
Thank you both. Hi, yes, my name is Mary, as I said before. Just give you a quick overview of the New York State Volunteer Program. Initially, we started in just the central and western New York region of the state. And a year and a half ago, we decided to develop a position that would oversee the entire state volunteer programming. And so now we've actually got, well, the vision, what we wanted to do was to be able to bring everything together, central programming, different areas of the state that didn't actually currently have a volunteer program, expand in those areas and develop new programs. Next slide, please. So this is actually our New York State Volunteer Programs team. And back to what Pippa was saying, we are broken up into different regions. So we have actually 6 volunteer coordinators across the state. Being with myself, I'm the volunteer programs manager, but I'm also still the volunteer coordinator for the central and western chapter or region of the state. And then we have on our Mashomick Preserve, we have a Becca Kusa who actually oversees the entire volunteer program on the state, which is a very large program in itself. We have a volunteer coordinator in New York State, New York City, Eastern New York, and the Adirondacks, and then on Long Island as well. So 6 of us, we meet every single month, and we go over different things that we've got going on. We're developing programming materials that again are centralized. We're also actually just got the word not too long ago that we can begin opening up and having volunteer events. So we can only have up to 10 folks, but we are very excited. We're excited about seeing everybody coming back, you know, just being a team. It's been a long time. So we're all very passionate about that. Another thing I wanted to say too is if you have any questions as you're going along through any of this, anything that has to do with volunteering, hours, liability forms, anything that has to do with that, please feel free to contact me at any time. I'd be more than happy to help you. So be on the lookout. Back to our volunteer, what do I want to say, not programs, events that we have. We just started scheduling them. So we do have 8 already that would be in the central and western and northern New York regions. So if you're interested in any of those, please also feel free to contact me and I can add you to the list so that you'll get that information.
Great. So now I'm going to dive a bit more into kind of how I fit into TNC and kind of provide some context for part of this project. So I'm a fisheries biologist and I work on our Great Lakes Sustainable Fisheries team. So I'm one of about 10 staff from New York all the way up to the northern part of Wisconsin, touching on Lake Superior. And we work basin-wide across the Great Lakes on fisheries. Next slide, please. So you might be wondering, okay, TNC, thought they were doing land, they're in the water too, so why the Great Lakes? Why have a fisheries program? So I'm sure there are probably numerous people on this call that love fishing, and as many of you are aware, we have a strong commercial heritage as well as recreational fisheries. Unfortunately, the salmon head got cut off in that upper left picture, but we have numerous awesome fishing opportunities. So fishing alone, while recreationally important, it's also a huge economic driver for this area. At one point, I was a few years old, but it's quoted to be like a $7 billion industry. And then we also, again, have that strong cultural heritage. And we also, as Brittany showed on that map, have really strong Indigenous peoples. So this area is really important. And it's also home to a number of people in the US as well as Canada. So it's an important region and I'm glad we're focusing on it. Next slide, please. So I will say that TNC, working in the Great Lakes, we couldn't do it without collaborating. We are one of numerous organizations that are involved and are only stronger by working together. So we work with state agencies, tribal fishery agencies, because the Great Lakes are binational, so Canada, US, we also work with the binational agencies as well as the feds, as well as various universities and research institutions. Next slide, please. So the focus of our program, not surprisingly, there are numerous fish species in the Great Lakes. So thinking strategically about how best to focus our efforts, we've kind of landed on what we call corygonines or corygonids. So these are cisco and lake whitefish. So cisco is the bottom left fish and lake whitefish is that upper right fish. And we've decided to focus on these species because of the important part that they play in the ecosystem. So if you think about our ecosystem and really kind of keeping it together and functioning in a healthy way, these species are key. So they provide important forage, or they serve as food for key predators like lake trout. They also help regulate nutrient dynamics in the lake. So we recognize that by working on these species, we can help lead to outcomes that will benefit the ecosystem. And I will say too, we're doing our second webinar where we will be giving a deeper dive into the target species. So this is kind of Cisco light, if you will, today. Next slide, please. So again, I just wanted to mention some of the history in the Great Lakes. So we actually had at one point the largest freshwater fishery in the world. So these are some photos from Lake Erie, and the landings were insane. You can see like these are both, I think, from Sandusky. So really help drive the economic viability of the Great Lakes region and its development. And to this day, we still have commercial fisheries for lake whitefish and cisco. Next slide, please. However, with all that fishing pressure, that alone has helped cause some declines, but you can't talk about native species in the Great Lakes without also thinking about aquatic invasive species. So we have in the upper left and the upper right are two fish species. So that's alewife in the left and rainbow smelt on the right. We also have sea lamprey on the bottom left. And then on the right, we have what we call dracaenid mussels. These are our zebra and quagga mussels. So together, these species have had a profound impact on the Great Lakes, affecting everything from like the fish themselves to nutrients, environmental conditions. So put this all together in one large blender, and our native fish like our corygonids, our ciscos, and whitefish have definitely been feeling the pressure. Next slide, please. So these are just some commercial harvest stats pulled from the Great Lakes Fisheries Commission. So commercial harvest isn't always the greatest proxy or indicator of abundance, but it gives you an idea of some historical context for what our landings look like over time. And I included Lake Erie here to show that this isn't something that's just unique to Lake Ontario. So here we have Lake Whitefish. So this is landings in Canada and the US. And you can see the records go back to 1867, which is pretty neat. But you can see the huge spikes in the commercial landings and then the declines. And actually in Lake Erie, we had a bit of not a comeback, but their populations were improving, but we're still at very low levels compared to where we were historically. And you can see the same trend in Lake Ontario as well. Next slide. And then when we look at Cisco, we see something similar. Again, what I think is really cool is mentioning that this was at one point the largest freshwater fishery. If you look at the scale on the left for pounds harvested, you can just see how much bigger it was. That was 45,000 at the top of scale there. So this was a big fishery. We had strong commercial pressure. And then you can see the late 1920s went down, little blip in the 40s, and then it's kind of flatlined. And I will say that cisco are now what we call extirpated from Lake Erie. So that means they're not found there, maybe a stray here and there. And in Lake Ontario, we saw that, again, that decline over time. And we actually don't have a fishery for them in New York. So that's partly why that line is flatlined. But they're still there. They're still in the lake. We're very interested in them. And we think that they can come back. Next slide, please. So you may recall the title slide, and it was tributary eDNA. So you may be wondering, what does all this have to do with tributaries? So a lot of our spawning for these fish happens in nearshore waters. So this is like 5 to 10 feet of water, kind of rocky habitats. But historically, we know that there are also some populations of these fish that actually went into the rivers, into these tributaries to spawn. And this was lost with time, or so we thought. So that upper left is a snapshot of actually a 2020 journal article that's finding that these spawning populations in these tributaries are actually coming back in Lake Michigan. So that's really exciting. From a nerdy fish bio, that's like, ooh, amazing. And in Lake Ontario, there was a big workshop in 2018 for Cisco thinking about research priorities. And this question was raised that if this is happening, and we know that there have been historical populations spawning in tributaries, like in Lake Michigan and Superior, could this also be happening in Lake Ontario? So that, from the native fish standpoint, is what we're really interested in. And we don't know. So we're hoping to hopefully find out more with this project. Over to you, Brittany.
Thank you so much, Pippa and Mary, for introducing everything so far. So Pippa kind of drew the connections for you on how the Great Lakes fisheries side and the invasive species really are interconnected, and how through collaboration we can start working together to move forward and learning more about these species. So I want to just do a brief introduction on the SLELO PRISM. I know some of you are very familiar with the SLELO PRISM on the line, and some of you are newer. So I apologize to those of you who are very familiar with the PRISM systems. But I just want to start by mentioning across New York State, we have 8 regional, these regional organizations that work together on invasive species issues. So here in our region, as I keep referring to as the Celilo region, we are on the boundaries of the Eastern Lake Ontario shoreline, the St. Lawrence River, and then also connected to the Adirondack Park, as well as connecting to the Finger Lakes and other parts of New York as well. So our mission is to kind of piggybacking on the Nature Conservancy, as we're hosted by them, but it is to protect native biodiversity and freshwater resources through our collaborative approach to invasive species management. So to achieve all of that, we focus in on some core programming and some really unique special initiatives. And so our core programming is based on prevention, early detection, rapid response, management, and of course, education and outreach. And so a couple of the initiatives that I want to briefly highlight is our macrophyte nutrient analysis. We're working with programs across the state to learn more about how our efforts to manage aquatic invasive species are impacting the ecosystems. We are conducting an aquatic restoration initiative, which is allowing us to actually do some management and restoration in a couple tributaries on the Eastern Lake Ontario shoreline. And if you're interested in that, you can visit our website for more information. In the SLELO PRISM, we've actually been doing a lot of environmental DNA monitoring over the last few years. And so that's kind of where this partnership really kind of built on. And then for a prevention program, our watercraft inspection steward program. I know this is like the super common invasion curve, but I really just want to highlight that we do have special initiatives and programs that are based on all of this. So basically the most cost-effective and easiest way to manage invasive species is by preventing them. So unfortunately, we can't prevent everything. And if some do get introduced, detecting those populations when they're in small numbers or very localized, we have more of an opportunity to better manage them, potentially eradicate them, or at least keep them in those localized regions. Unfortunately, as you start getting the areas of infestation increased and populations increased, it turns into more long-term management, and it's a little bit more costly and a little bit more work as well. So the eDNA aspect of this is detecting species early on. So we're in that, okay, potentially we might be detecting some invasive species in small numbers. And, from the invasive world, we want to potentially try to manage to eradicate. However, with the native species, that early detection will also allow a further response. In SLELO, we have a huge region that we work in, and basically, I just want to give a quick breakdown on how we decide where to work and what projects we work on. So we have a formal process for creating these priority conservation areas, and there's some information on the screen that you can see here, but basically these are areas that are really important to protect, and I'll show you a list of those areas in a moment. And then also we focus in on certain species. So I don't know if you know this, but there's almost 200 invasive species that have been introduced and established to Lake Ontario or the Great Lakes. And so it's a lot of species to look at. In the past, we were nominating and working with partners, but now we're kind of working on this new tiered system where basically it's more focused on populations, the actual impact of the species based on, the negative impacts that they might have, and then also the difficulty of eradication or cost to control. So this is a longer list. And again, if you would like to learn more about our prioritized species based on this tier list, you can visit our website, and we have pages for all of these species with more info. So I'm not going to go through everything. The species that are listed in blue are the aquatic species, and we will actually be focusing on some of them for this project, but I'm not going to give any spoilers, so I'm not going to talk too much about that. These tier 1 species on the left-hand side are those that are not actually in SLELO yet, but we're hoping to really catch those, any species or any individuals that get introduced as early as possible. So talking about the prevention side of things, we do have a watercraft inspection steward program. We basically, you can see on the map on the left-hand side, all those dark blue dots are locations where our stewards are actually at launches, working with boaters, making sure that we're trying to prevent the spread of invasive species to or from waters. And we have learned over the past couple of years how much people are actually traveling across New York State from launches, between launches. So it's definitely a really good program. This past year, even with COVID, the pandemic, we were able to talk to almost 30,000 people. So it's a pretty big program. It's very exciting to try to at least keep the prevention efforts. We did intercept over 1,200 invasive species on boats and trailers. The priority conservation areas that I mentioned to you already are listed here on that right-hand side. And so these were areas that through our partnership were determined to be really important to protect. And so myself, you can see an image here, and my colleague Robert, we're the aquatic and terrestrial coordinators. And we do early detection and field surveying at all of these sites on a every other year basis. So we go out and do monitoring both in the aquatic ecosystems and on land. So just a brief mention of our restoration work. So one of the major projects that I'm working on is our Aquatic Restoration Initiative, and we will be working in Lakeview Wildlife Management Area and Deer Creek Marsh Wildlife Management Area. And so in those two areas, we're doing some management for invasives and also going to do some planting of native species and trying to improve these habitats for the long term. So we completed the first year of that, and now we are on year 2, and our management actually starts in a couple weeks. So if you are in any of those areas and you see some work happening, stop by, say hello, and I'm sure our contractors will be happy to answer questions about what they're doing. So I know I just went through a lot of information, but as I hope that you're now aware, we have extensive efforts underway for both preventing the spread of invasive species, managing their populations, doing some restoration work, and then also protecting our native habitats and wildlife. And so through all of these efforts, we would not be able to do this without collaboration, which is essential to actually limit any unintended negative impacts that may be continuing to destabilize these systems, thinking back to what Pippa mentioned, so even as big as climate change. So our eDNA work might seem like they're just small scale projects. However, we can have really large impacts on the ecological integrity and resilience of some of these landscapes. And I know you're looking at the map here like, okay, the Algonquin to Adirondacks, I'm interested. But this region actually represents one of the last intact north to south forest corridors in the Eastern North American region. And so it provides a really important habitat for mammals, migratory birds, and a lot of other organisms that move across the boundaries, because I actually did see that we have some people from Canada on the line as well. So shout out to you guys. You know, we have these borders between our countries. However, nature knows no boundaries. So we are anchored between these two major parks that have huge, vast areas of protected lands and then multiple areas in between. And so the Algonquin to Adirondacks is a really important part of our work as we try to improve this connectivity for wildlife. So to kind of build on that a little bit, the A2A region, or the Algonquin to Adirondack region, was actually inspired by a 700-pound moose named Alice in 1998. She was actually spotted and reported multiple times as she traveled through Oswego County in the Celilo region. You can see the dark blue circle down here, and she was headed west. And DEC actually ended up capturing her almost in Rochester, and they put a collar on her. They relocated her to the Adirondacks. So also shout out to our Adirondack partners on the line. So they moved her to the Adirondacks, where she lived for a couple of years. And then something about moving out west for her, she decided to go northwest this time. And with her collar, they actually tracked her traveling 355 miles up to the Algonquin Park. And you can see the red line here is actually the way that she traveled. So the Thousand Islands acted as a skipping stones, so to speak, for her to travel across the river. And so as we're going to transition to introducing more of the project next, I just wanted to kind of remind you of the story of Alice, the moose, the 700-pound moose moving around, and just really kind of bring into home the connectivity of our land and our regions, and why, although we're only doing some small work in some of the tributaries along Eastern Lake Ontario and in the St. Lawrence, all of the work that we're doing can have a large impact, and your involvement and participation, even in just these webinars, is very important. So thank you, and I will let Pippa take over.
Thanks, Brittany. So now we're just going to jump into a little bit more detail on our project. So you've heard from me about our native fish and the potential to possibly be using tributaries for spawning. And you've heard from Brittany the importance of early detection and monitoring. So with this in mind, we kind of have these two supplementary projects. And what's at the core of them is very first is to determine presence of these species. And this is where this novel technique of using what we call eDNA, environmental DNA, is something that caught both of our eyes, and we realized that it could be a way to bring our programs together. So our aim is to basically collect water samples and run them for this eDNA in select tributaries along Eastern Lake Ontario and the St. Lawrence to assess presence of our corygonids, so it's the ciscos and the whitefish, during their spawning season, as well as to determine what other invasive species might be in the area so that we can help with the efforts for the SLELO PRISM. So we're really excited about that. Next slide, please. So here are actually some of the tributaries that we are hoping to get out and sample. So the ones in orange are all of our sites, and the pink are just ones that I've called out specifically for lake whitefish and cisco based on some historic records that I've found. And so I did mention that we'd be wanting to detect presence of cisco and whitefish during their spawning season. So they're known to spawn from November to December. So we're hoping to kick off sampling in August and run it all the way through December. So that way we can get a nice chunk of time to capture the invasive species as well as our hopefully native species, fingers crossed. Next slide, please. Over to you, Brittany.
Yes, thank you. So with that, I just want to talk a little bit more about the project and kind of, you see our locations, our spread is literally from Oswego all the way up to Cornwall Island, up near Messina or past Messina. So we have this huge range that we're working in, and Pippa and I are the co-leads on this project. So within the Nature Conservancy and SLELO. And then we also are working closely with our education outreach coordinator, Megan, and Mary Ripka, as you heard her introduce herself on the line. We are hiring a seasonal tech to assist us with this project. So hopefully by the third webinar, if anyone's joining us in that August webinar, we'll have our seasonal tech on the line who will be working more closely with the volunteers out in the field. And I do want to just mention something that's super awesome about this project is that in the past, we've always done most of our work either on a table in a parking lot or on the back of the boat in camping chairs. And one of the coolest things about the project this year is that we're actually in process of purchasing and building out a mobile eDNA lab. And so basically this is an enclosed cargo trailer that will have everything we need to do to process all of our samples that we've collected in the waters. And so if anyone volunteers with us or joins us for any sample collections, you'll get a chance to see our new lab as we work through all the kinks of trying to purchase something during a pandemic and also learning how to create our own mobile lab. So it's super exciting, and we would not be able to do this project without some of our key partners. I know, and I apologize if there's anyone else on the line that is not specifically called out yet, but I just want to mention the USGS, US Fish and Wildlife Service, New York State DEC, New York Sea Grant, SUNY Oswego, SUNY ESF at the Thousand Islands Biological Field Station, Cornell University, and the St. Regis Mohawk Tribe. Without our partnerships and collaboration, a project like this at this scale across the entire region would just not be successful. And another super big shout out to the volunteers on the line. I'm familiar with some of you who I have met and some of you who I haven't. And with this project, we are seeking volunteers to assist us with setting up the mobile lab, getting things together for that, actually going out and collecting water samples, transporting water samples. If anyone has photography skills, capturing photos or videos. We have a super cool new underwater remote operated vehicle that we'll be using this year, which is basically you use a remote control system to drive a little device under the water with cameras so that we can see what's under the water as well. And then also, we seek anyone on the line to assist us with information sharing. So sharing information about the project, about our programs, and about our work. And we'll do the same as well. Or if you see this list and you say, hey, I actually have something that I can contribute that you haven't listed, again, please feel free to contact us, and we're happy to work with anyone just to improve this program and make it as collaborative as possible.
So we wouldn't really be able to launch this work at the scale that we're planning if it weren't for the Archonic Foundation. So they were really gracious and gave us an awesome grant that will help us not only purchase that trailer, but help with our sampling and our processing of the samples. So I just wanted to give a shout out to them, as well as give you guys a little bit more information on who they are. So they are located in Messina, New York. So that was kind of that northern part of the St. Lawrence where some of our sites are. And really what they do is they're kind of, they're associated with Archonic, which is a large industrial company. And the foundation likes to partner with nonprofit and community organizations to help the community by enhancing education through skill building learning experiences. And they like to promote environmental sustainability and advance social equity. So they're an independently endowed foundation that have assets of about 180 million, and they invest this back into the community. So we're just really thankful that they saw the value in our project and the type of outcomes that we're going for. Fortunately, they couldn't be on the webinar with us today, but if they watched us afterwards, thank you again. And I think we've both kind of mentioned our next webinar. So this was the first webinar in a series of 3. So if you're interested, please join us for our second webinar. This is where we'll go into some more specifics on what eDNA is, as well as the target species. So we'll tell you a bit more about the project on this call. And it's scheduled right now for Wednesday, June 16th, also at noon. And here's the website, that's SLELOinvasives.org/eDNA if you'd like some further information. So that is it for us. We're more than happy to take any questions that you guys might have. You can either use the chat function, type R in the box, or there is that little hand raising option, which I always find a little bit awkward. Never know when it goes back down, but if you want to, you can put it up. But yeah, we're more than happy to answer any questions that you may have. Okay, so I see one in the chat of how many species will you be testing for? So we will be going into this a bit more in our second webinar, but we will be testing for some of our native species. So that would be the cisco and whitefish, and we're thinking about 1 or 2 native species as well. And I'll let Brittany speak to the invasive species, but right now we're also in it's a bit of a juggle, because with COVID, it's impacted everything from being able to get the supplies to run the samples. So we're still working on making sure that there's actually what they call the assays, which is what you look for when you run the samples for different species. But in the end, we're really hoping to probably have about 10 to 12 species tested for on our panel. Don't know, Brittany, if you want to speak to any of the invasive.
Yeah, no, I mean, that's a pretty good description. So we're hoping to include the Asian carp group, northern snakehead, a newer one that some of you might not be familiar with, the Asian swamp eel. We're also looking at potentially the rusty crayfish and tube-nosed goby, some of the species that might be impacting our native forage fish group as well. So yeah.
Okay, looks like we have a question from Rob. Will you be using qPCR, and what lab will support the project? So yes, so if you are at all familiar with eDNA testing methods, there's kind of 2 ways. There's the specific, this is what we're looking for, go out in the water and see if we can find it, and that is done using qPCR. Or there's something called like next gen or high throughput, where you just run your water through and you basically test it against the library. So we are opting to use qPCR. It's a very effective method, and it's been used in numerous studies of this type across the Great Lakes and elsewhere. At this time, we are still working on getting our lab aligned. We have 2 in New York that we would like to work with, but as I mentioned, it's really been interesting to see the impacts of COVID trickle down. It's everything from getting the reagents in the lab to them having the capacity. So that's why it's kind of a little bit of a juggle right now, but we will have a lab and we will have samples. Otherwise I'll die out my tributary, making sure.
Yeah, it's definitely been an adventure for Pippa and I, moving through the pandemic and doing this project. So as she mentioned, so with actually COVID testing, most tests are actually ran through qPCR. So most of the lab equipment that we would need or would typically utilize is very hard to get. But yeah, so I see the next question asking if it's possible to wear multiple hats while you're a volunteer, and absolutely. Unfortunately, I think most of us have learned how to juggle multiple hats at the same time. So yeah, I mean, there's so much that we can be doing out there and kind of doing it all in collaborative. So that's absolutely, if you're interested, we're happy to have that. If anyone else has questions or comments or anything else you'd like to ask. Oh yeah, so time commitment for volunteering. And honestly, that is totally up to each person. So if you are We actually are keeping track of everyone on the line. So technically your volunteering time commitment starts now. But basically whatever you're willing to contribute or want to contribute, I think that for most people, once they get out there and start doing their first water sampling, they might have only had the intention of joining us once, but then want to join every time because it's pretty fun. You get out, it's pretty conversational. You get to know each other and just learn about the work that we're doing. So time commitment is anything that you can provide. Yes, give me 1 second, and the St. Lawrence County Planning. I'll pull that map back up with our sites. So as you can see here, if you look down to the left-hand side, we are anticipating starting in the Oswego River and up Eastern Lake Ontario, and then all the way up into the St. Lawrence as well.
So Brittany and Pippa, if I may, I just want to, compliment you guys. This is a great project, and I'm really impressed. I'm impressed with the scale of the project, not just in sampling sites, but the scale of the collaboration that you guys are putting together with the various partnerships. I think it's really important work, and from what I see from this. Great teamwork, great collaboration, and best of luck moving this one forward.
Thank you.
Thanks, Rob. Guess while we're on this, I'm waiting for the next question too, just to speak to some of these sites. We are planning on sampling in the habitats where we expect the species to be. So that's part of like why some of these are pink. It's again pulling from like historical data, but also knowing that in Lake Michigan for like whitefish, for example, they're found to use some of the similar habitats to walleye, for example. So we've done some of the background research on where we think some of these kind of primo habitats to sample in would be.
Yeah, and that's definitely where our partnership comes in as well. We have everybody works on different things or focuses on different initiatives. So we've been able to utilize the knowledge of our partners as well, who we've been working with to try to hone in on these locations. And some of them will be accessible by boat, some of them are accessible by land, and we're working on the field verification of that. Basically, we're just juggling all these pieces as they all fall into place in the meantime.
So we have a question here of when does sampling / volunteering / field work start? So we are looking to start our field work in August. And so we're actually planning to do a volunteer specific webinar in August, as well as some trainings. And again, that will run through December. The native fish like to spawn in the winter. So, so November and December. So some of our efforts will pick up then to make sure that if they are possibly there, we can increase our probabilities of detecting them. Oh, we have a question of what are carrigonines. So carrigonines is like this kind of general term that includes cisco, whitefish, and there's actually like several species of cisco. We have some deep water ciscos like blackfin. So it's kind of a more broad term for them. And fun fact, they're a member of the salmon family, in case no one knew that.
Awesome. We'll give everyone another minute or so to, if anyone has any more questions. Awesome. Well, I think it seems that there has been a lull in questions, so I will take that opportunity to just thank everybody for joining us today. Actually, I do see one more question, so I'll answer that before I continue. Thanks, John. And so actually, we have all of the equipment that would be used for sampling. So basically, all you would need to bring is yourself and potentially some, sunblock or whatever else you might need, bug spray, that kind of stuff. So pretty much we've got everything for the sampling that you would need. But so with that, I just want to say thank you to everyone who joined us today. And the next presentation on June 16th, we'll go into a lot more detail about the different species, the sampling locations, and just kind of hone in on the project a little bit more. We didn't want to take too much time, each day. I know sometimes a 2-hour webinar can get pretty exhausting. So we just wanted to split them up for everyone to join us at multiple times. So thank you so much. And make sure that if you haven't already, register for that second webinar. And feel free to reach out to us if you have any more questions or want to volunteer.